Paramutation is a type of transcriptional level gene silencing mechanism. Hello and welcome to Dr. Varsha Gaitonde's channel. Friends, in this lecture we shall discuss about paramutation and its some relation with gene silencing. Gene silencing is a technique that aims to reduce or eliminate the production of protein from its corresponding gene. There are two terms, one is knockdown and one is knockout. Gene silencing basically works with knockdown principle rather than knockout. So let us see what is knockout and knockdown mechanism. When genes are knocked down, their expression is reduced, means complete elimination of the expression is not preferred here, whereas in contrast when genes are knocked out, they are completely erased from the organism's genome and thus have no expression. As you know, in case of operon concept, there are always switch on and switch off of the mechanisms. Okay, switch on for the production of particular product and switch off to get rid of that particular product. The same principle works out here also. So the process of gene silencing is not a new concept. It was there before 1900s also, but the progress was quite low in the research field. But the success was achieved in the 1998 when Andrew Fryer and Craig Mello worked with RNA interference technology and they were awarded with Nobel Prize in the year 2006. So as you know, there are two types of gene silencing in broad terms. One is transcriptional gene silencing, then post-transcriptional gene silencing. Briefly, we shall see what is the difference between these two. In case of transcriptional gene silencing, promoter is silenced. As you know, in the operon concept, you will be having certain regulator genes, then you will be having promoter, operator, then structural genes, and all those things. Okay, so here you see in the transcriptional gene silencing process, promoter is silenced. Okay, then genes hypermethylated in promoter region. Methylation is an epigenetic process where activation of particular genes is regulated. Hypermethylation means the any particular product's activation or production is reduced, that is hypermethylated. Purpose is viral immunity. Similarly, post-transcriptional gene silencing mechanism is related with promoter activation. Here genes hypermethylated in coding region. The purpose is viral immunity. Another kind of gene silencing is also there, that is meiotic gene silencing. Just to recap all the types of transcriptional, post-transcriptional and meiotic transcriptions we shall name. In transcriptional gene silencing, genomic imprinting, paramutation, transposon silencing or histone modification, transgene silencing, position effect, RNA-directed DNA methylation. These are the types of transcriptional type of gene silencing. In post-transcriptional type, RNA interference or RNAi, RNA silencing, nonsense mediated decay. The third is meiotic gene silencing type. Here transvection and meiotic silencing of unpaired DNA. So in this lecture we shall only restrict our discussion with paramutation. In case of transcriptional gene silencing, why I am taking definition? Because paramutation is a type of transcriptional gene silencing. It is a result of histone modifications creating an environment of heterochromatin around a gene that makes it inaccessible for transcription machinery, maybe with the help of RNA polymerase, transcription factors, etc. Okay, here what will happen? Histone modification. Where you will find the histones? In case of chromosomes, you will find DNA. For DNA binding, there you will find the histones. What will happen? It will create such an environment that heterochromatin around a gene that makes inaccessible to transcriptional machinery. What do you mean by transcription? It is making the RNA from DNA. This machinery will be stopped with the help of certain mechanisms. So, if we particularly talk about paramutation, there are certain terminologies. What is paramutation? It is an allelic interaction in which one allele referred to as paramutagenic causes a heritable change in the expression of a homologous paramutable allele. There are again two terms, one is heritable and one is transferable. Heritable means the characters inherited from a progeny by a progeny from its parents, okay, and transmission means from one organism the characters or any genes transferable between the two individuals as we do it in the transgenics. Paramutation is an epigenetic phenomena. Epigenetic means there will be surface level changes but no change at the DNA level, okay. Paramutation is an inheritable change in gene expression where allelic interaction takes place. Here, three or four terms are very important. One is paramutagenic. Genic means causing. Okay, the alleles having ability to silence other allele. That means I am having ability to control some of the pupil. If I am taking some class to primary school children, so what I am having, I am a teacher and I am having ability to make them to keep quiet. Okay, that is a kind of paramutagenic. Just an example to mutagenic kind. Okay, so paramutable, here the children are paramutable, that means they are able to be silenced. Okay, then paramutated means once they get silenced they can be called as paramutated. The newly silenced allele remains stable in the next generation even after segregation from the paramutagenic allele and acquires paramutagenic ability itself. These three terms are very important: paramutagenic, paramutable, paramutated. Then comes neutral alleles. The alleles unaffected by exposure to a paramutagenic allele are called neutral alleles. That means if somebody is asking you to keep quiet also, you are not ready to do that. That is nothing but neutral allele. Means whatever you want, you are doing. Term paramutation is taken from a Greek word. Para meaning beside or near, beyond, aside. As you know, mutation meaning means sudden heritable change. How paramutation is different from mutation? Mutation is sudden heritable change. Some external factors are affecting and because of that mutation is happening. It may be spontaneous mutation or induced mutation, but in case of paramutation the work is with the alleles, okay. The definition is paramutation describes the occurrence of meiotically heritable epigenetic changes in gene regulation and trans-inactivation behavior. The behavior are recognized by non-Mendelian inheritance patterns that are found independently of chromosome transmission ratio, distortion, and at least in plants in the absence of parent of origin effects. Coming to the characters of paramutation. Paramutation as it is directed, heritable, where one allele at heterozygous state affects the other allele. Here, heritability, direction, methylation, heterozygosity, allelic interaction, all these things you should remember. The newly silenced allele can further silence new targets. Allelism is not a strict requirement for paramutation. Here the meaning is if I am silencing you or asking you to keep quiet, you are also asking somebody to keep quiet. That means you acquired that character from me or you just taken that character from me. Okay. Allelism is not a strict requirement for paramutation. Paramutation is associated with the DNA methylation changes. The concept was given in maize for the first time by Alexander Brink in the year 1956. Like that, Stam and co-workers in 2002 also worked on paramutation in maize, provides convenience evidence that chromatin level regulation underlines the phenotypic differences between alleles that participate in paramutation. Genetic analysis implicate that small RNA or sRNAs and RNAi mechanisms in affecting paramutations operating in both plants and model metazoans. So there are four popular examples for paramutation. Two are in maize, one is in mouse, and one is in Arabidopsis. Apart from this, in some other organisms also the mechanism is observed, but these are the popular one and easily understandable one. In maize, paramutable capital B1 allele is silenced by the paramutagenic B9 allele in the first generation. In the next generation, newly silenced B1 allele is paramutagenic and silences a naive B1 allele. Okay, this is the figure. In this figure, you may find that there are two alleles or genes responsible for dark and light color. Okay, B1 is dark colored and B dash is light colored. When these two are crossed or these two genes are present in two different parents, what will happen? We'll get the F1 which is having light pigmentation. Okay, this is all related with anthocyanin pigmentation. So light pigmentation is obtained because here the B dash is working on B1. That means this B dash is paramutagenic and B1 is paramutable. Okay, so again when F1 is crossed with B1 which is a dark one, again it gets silenced and it will give the light anthocyanin pigmentation. Like that, again in maize, paramutable RR, where standard allele is expressed in the first generation when heterozygous with the paramutagenic R stripped. When the heterozygote is crossed to a plant homozygous for a neutral R allele, RR standard is silenced in the second generation progeny. Again, this is an example where RR, which is a heterozygous showing dark color, which is crossed with light color stripped one, what you will get? You will get the dark one. Okay, because dark one is paramutagenic here. Then again, if you are crossing it with the recessive homozygous one, what you will get? There will be segregation, light one and R stripped, but both are light one only, but characters expression is different. Rest 2 I'll just read out, I am not going to explain it. In mouse, the chimeric gene the allele when crossed as male activates the maternal expression of normally paternally expressed allele, resulting in biallelic expression in the first generation. Maternal expression maintained when the chimeric allele is segregated away in the second generation. Okay, this is the example where the paternal two characters are crossed, then biallelic F1 is obtained, and again when it is backcrossed with the paternal one, what you will get? You will get the segregation in biallelic and maternal. That means the two paternal characters are activating the maternal character. And the fourth example is in Arabidopsis autotetraploids, where cross of hygromycin resistant plants with hygromycin sensitive plants carrying silent transgene produces first generation progeny that are hygromycin resistant. Self-fertilization of the hybrids produce mainly hygromycin sensitive progeny. Okay. The last figure, this one. What will happen? Resistant and sensitive are crossed, and in F1 you got resistant, and in F2 when it is selfed, what you got? You'll get more of sensitive kind. Okay, now clearly the experiment of an Alexander Brink, with what he worked with the two genes in maize. One was booster B and another was red. Both are recessive genes. Both genes encode transcription factors that regulate synthesis of anthocyanin pigment. At both loci, paramutable alleles have higher expression than paramutagenic allele, but several observations suggest that different mechanisms control these two loci. They differ in genetic behavior, molecular organization of the loci, imposition of epigenetic work like DNA methylation and altered chromatin structure. At B, two alleles participate in paramutation. When the paramutable B1 allele or dark pigmentation is crossed with paramutagenic B dash, the F1 progeny exhibits B dash phenotype. That means this B1 and B dash are the two alleles of recessive B gene. Okay. This phenotype is stable and the new B dash allele is paramutagenic such that crossing the F1 with the new naive B1 leads to production of only B dash phenotypes. See, this is naive B1. This when the dark one is like this. Okay, to explain you in better way, this got paramutated. Okay, this when again back crossed or crossed with a normal dark B1, again you got the light one. That is what is explained here. At the molecular level, B locus maintains single coding region. Reduced pigmentation of B dash is because of reduced transcription. There are no differences in DNA methylation or DNA sequence in the coding region of paramutagenic B dash and paramutable B1 alleles. This sentence is again very important. So let's see the significance of paramutation. The inheritance behavior of paramutation alleles permit fast diffusion of a particular gene expression or level phenotype in a population, even in the absence of other evolutionary influences, thus breaking the Hardy-Weinberg law. What this Hardy-Weinberg law states, in a random mating population, there won't be any changes whenever the natural factors like mutation, migration, random genetic drift, selection are absent. Okay, so this is the deviation from Hardy-Weinberg law, and you know that Hardy-Weinberg works on the population, means it is a principle applicable at population level. As with other gene silencing phenomena such as quelling in the fungus Neurospora crassa, transfection in Drosophila, co-suppression and viral induced gene silencing or VIX, described in transgenic plants and RNAi in the nematode C. elegans, paramutation occurs without changes in the DNA sequence. That means it is an epigenetic phenomenon. Molecular basis of paramutation is not fully understood till now. The involvement of RNA causing changes in the DNA, methylation and chromatin structure of the silenced genes. Next, there is some interconnection between different concepts also, like paramutation and genomic imprinting. As what is genomic imprinting? It refers to differential expression of maternally versus paternally inherited alleles. Imprinting has been reported in many mammals. That means paternal character expression and maternal character expression won't combine. They express differently in the progeny is nothing but genomic imprinting. Work on mouse suggested a connection between genomic imprinting and paramutation and may provide some clues about how maternally imprinting genes are regulated. Next popular and famous example is paramutation and polyploidy observed in wheat. Paramutation like regulation as an important determinant in controlling gene expression during changes in ploidy level. There is accumulating evidence that the interaction between two different genomes trigger epigenetic changes in allopolyploids. In wheat, both upregulation and downregulation in gene expression were found. Some changes were specific to one of the parental genome, and gene silencing was correlated with increased DNA methylation at the affected loci. Changes in DNA methylation and gene expression are also seen in synthetic Arabidopsis. In one study, 0.4% of 700 genes analyzed were transcriptionally silenced. Thus, changes in gene expression may explain typical phenotype that arise when plants undergo a change in the ploidy. Coming to the utility of paramutation. Emerging importance of chromatin states not only as heritable but also as transferable. That means this is utilized in transgenic technology. Allele specific communication may be mediated by non-coding RNAs that catalyze trans-acting alterations in chromatin structure that is in turn control expression state. Mechanism for establishing paramutation are likely to vary among different systems, but maintaining stable expression states appear to be modulated by a common network of chromatin level regulators. Okay, as you know, if we especially talk about plant breeding perspective, you know maize is a quickly changing crop because there are certain principles like paramutation and all where the things will be changing. So to keep the hybridity, or for example I have developed one hybrid and I want to maintain it for a longer time, maybe I want to keep its integrity for 10 years, 20 years, like that, and I want to maintain their inbred germplasm. So I should avoid all these things. Before that, I should see the quantity or the quantum of these mechanisms in that particular um, inbred, because as you know there will be minute variation between the genomes of inbreds also. So in those cases paramutation is helpful. So friends, so far we discussed about gene silencing, paramutation, scientists contribution, Nobel Prize, then the different experiments and the proofs for paramutation, its utility, and all those things. If you have any doubt, please keep it in the comment section. I have given some references also. Please do not forget to share, subscribe, and like the channel and video. Thank you.